Good afternoon, and welcome to What Genetic Testing Reveals, the benefits of genetic testing for children with autism spectrum disorders. This session is being presented by Dr. Richard Boles from NeurAbilities. This webinar is one of many that we at the Family Resource Network have in our webinar series, so I urge you all to go to our website, familyresourcenetwork.org, to find out more information about our upcoming webinars. One of our programs that we would like to promote is our Health Insurance Navigator program, which in this day and age, we usually do in-person meetings with individuals that are seeking health insurance, but we are now doing virtual meetings. So if you're interested in finding out more information about health insurance and any types of insurance that you might be eligible for, please contact us, and the contact information is on your screen right now. In today's webinar, you all have a control panel on your screen. You can click the orange arrow to open and hide your control panel. Under the audio tab, you can pick mic and speakers to use your computer, or choose the telephone option if you're listening over the phone. All the attendees will be on mute during this webinar, but if you have any questions, you can type them into the question section of your control panel, and if time allows, I will read those aloud to Dr. Boles at the end of the webinar. This webinar will be recorded, and we will post that on our webinar archive on our website. In addition to that, you'll also be receiving a certificate tomorrow in your email that you have completed this webinar. And also, you will be receiving an evaluation. We really value your opinion in this evaluation, so please fill it out and let us know some ideas that you might have for upcoming webinars. Dr. Richard G. Boles is a medical geneticist and a pediatrician who specializes in mitochondrial medicine, functional disease, and autism spectrum disorders. His expertise stems from decades of both clinical work and research at a major academic center, as well as from his most recent experience in cutting-edge biotechnology and genomics. He uses an innovative and integrative approach in both diagnosis and treatment to best serve his patients. Dr. Boles leads the program at NeurAbilities Healthcare, which is currently available for physicians and providers accessing our peer-to-peer service, as well as patients accessing direct-to-patient service. Physicians and their healthcare providers will be able to consult with Dr. Boles directly about reanalyzing previously done genetic testing results, advice for managing neurometabolic disorders, or to obtain guidance on choosing and interpreting genetic testing. Neurogenomics can help to unravel the biological causes and contributions for many diseases and disorders, as well as provide important information for changing clinical management, including mitochondrial disease and dysfunction, chronic pain, epilepsy seizures, unexplained and undiagnosed disorders, and much more. Dr. Boles attended Yale University for medical genetics and metabolic disease. And Dr. Boles is also part of the NeurAbilities organization. He is actually based out of California, so it's a little bit earlier for him over there, but I know this webinar does have a national scope, so I'm sure people are logging in from all over the nation right now. So at this point, I want to introduce Dr. Richard Boles, What Genetic Testing Reveals.
Hello, am I on right now?
Yes, you are.
Okay, great.
And I can fine.
The slide is fine?
Yes, perfect.
Okay, so I'm going to try to connect the dots to some degree between the genetic sequence, which you can see as a series of letters on there on the screen, and then the behavior of autism. And there is a lot of steps in between. And it is extremely complicated, but I'm going to try to, more than anything, try to show you what the power of genetic testing can do right now, and why you should consider genetic testing for your child, but also that not all genetic testing is the same, and that really you would need to have some of the more modern testing to get much better results. And why is not the slide progressing? There it goes. So here's my disclosures, but who I am. I mean, you've already heard about some of that. I was at Children's Hospital Los Angeles and at USC as a faculty member, a professor for 20 years. So I come out of academia. And right now I'm in private practice. My primary physician responsibility is now at NeurAbilities, which is based in New Jersey, but really has a national scope. I also have a small private practice in Pasadena, California, which I usually see patients in person, but now it's all telemedicine. I'm the medical and scientific officer, the chief officer of NeuroNeeds, which is a company that produces some products that is used to treat autism based upon the genetic findings that are frequently seen in kids with autism. I have been involved as the medical director of several different companies, including 2 DNA sequencing companies. So that's where I got a lot of this expertise in reading the DNA sequences. I do expert legal cases, particularly medical child abuse. And then I've mentioned before, as I was a professor, I had 20 years of research experience and still do some of that. Here are some other caveats. There are no official, looking at the bottom, the blue one, there's no official recommendations for genetic testing in neurodevelopmental disorders. Autism is one of many neurodevelopmental disorders, ADHD, developmental delay, epilepsy, there's many more of them, but autism is just one of that part. There used to be some recommendations, but they are so many years old now that they're completely immaterial. So it's at this point right now, this is basically what my own opinion is. And having been a clinician, which is my main role right now as a physician of these patients, and also having been involved in sequencing companies and in research. The other thing is that if you look at the green at the upper left, autism is now, like everything else, has become personalized medicine. If you can do the entire DNA sequence, is what we do most of the time now, in most of the patients that do whole genome sequencing, then you can find out the individual cause of autism in that patient. Autism is a symptom. There are many, many different ways in which you can get there. And when you find the individual cause of autism, then there's often going to be an individual treatment. And there's one patient that has that particular cause, and the treatment is based upon the individual genetics. And that's really important because it's not like there's thousands of patients and that there's research studies that have been done and that you have drugs that are FDA approved. So this is personalized medicine to determine the cause of the disease in individuals. And of course, there's no way that you can do studies on large numbers of people and to be able to get FDA approval for that. So all of the drugs and treatments I'm going to be talking about are off-label. The other thing is that I am the director of the Neurogenomics Program at NeurAbilities, and so I'm going to be telling you about genetic testing that we can actually do. So there's some sort of a conflict of interest, and then the upper right warning is at you're at risk for this talk. Okay, the first patient I'm going to talk about is Carter. Carter was 1 year old, and following a vaccination, he stopped developing speech for the next year. Unfortunately, I have more than a few patients that had autism that started after a vaccination. He was later diagnosed with autism. Biochemical testing suggested the possibility of a mitochondrial disorder. And by the way, about 60 to 80% of patients on the autism spectrum have biochemical testing that suggests that there's mitochondrial abnormalities. That doesn't mean that mitochondrial disease is the cause of most autism, but there is a degree of mitochondrial dysfunction in most of the patients. And then DNA sequencing revealed a specific mutation in the TRAP1 gene. I had noted that several other patients had had the same variant, and those patients generally had what we call functional symptoms, such as chronic pain, fatigue, and GI dysmotility. This gene encodes for a chaperone, you can see from the top of there here, that is important in oxidative stress, in protecting the mitochondria against reactive oxygen species. The connection between autism and functional disease is really how I got involved in it. I started off in functional disease, cyclic vomiting syndrome, irritable bowel, migraine, chronic fatigue syndrome, depression, and all of those disorders. And then I noticed that a lot of my patients had autism, and more importantly, a lot of their brothers had autism, and that there must be a genetic connection. So a lot of the patients with functional disease had this variant in the gene. And this on the lower right-hand side, it's a little cartoon as to what the TRAP1 gene looks like. And then you can see in the green on the bottom there, that's ATP. That's the molecule of energy. So ATP is broken in that this molecule acts like a claw. The claw is now the claw is at the top of the molecule in the cartoon, and the claw is now closed in this particular one. But the ATP operates the energy to open and close this claw, which then grabs proteins and protects them in an oxidative environment, just by pretty much just wrapping them up and protecting them against the environment from unraveling. Unraveling is denaturizing or cooking. That's what happens when you cook food, is you unravel proteins. By molecular modeling, and this was by colleagues at Georgia Tech, ran their computer system an entire weekend to find every single drug and every single every oral supplement, dietary supplement, anything they can find that can go into this ATP binding site at the bottom and stop the molecule from being, the mutant molecule from causing its trouble. The mutant molecule being the one with the mutation. And it found that Chitril, which is ganastatron, is a drug that's often used for chemotherapy-induced vomiting and nausea, that it fits into the binding pocket really well and might work. So we tried that in Carter, and you can see the differences in green here. The ones that have a little star on it were ones in which, when the drug was removed, he got worse on, and when the drug was put on, he got better on, over and over again. He got dramatically different on this drug. In fact, that he went from running around and causing trouble, and there's no way they could ever take him to a restaurant or anything like that. And when they gave him the drug, he was able to sit down at a restaurant and eat and behave himself. So it really made a dramatic difference. And it is you can imagine the insurance company would not consider this to be an approved use of the drug. So they denied it. It's an expensive drug. And so he went off of it a couple of times at that point. And then later, the parents decided that it was so important for his behavior, they decided to pay for it anyway. And they finally got the insurance company to pay for it. Then they went through 2 nationwide shortages of the drug. So he went off of it, to my knowledge, 4 different times, and he did get worse each time and then got better when he went back on. So it really is the drug making a difference. And this can show you that core areas of autism, behavior, language, and social ability can improve if you know the underlying cause of autism. It is true there's no approved treatment out there that in general, for kids with autism in general, will improve those core areas. But that's because everybody with autism has a different cause of disease, and there's nothing that can make all of them better. You really need to know what the cause of autism is in an individual, and then if you can treat that, not everything can be treated, you can improve the core symptoms if you're so lucky as to find the cause and to be able and have a cause that can be dealt with current technology and understanding. The next one here is Pam. So I showed you a child, here's a young adult. And you can see at the beginning, I say relatively uncomplicated. There's no such thing as uncomplicated autism. Everybody is unique individual, but these two have fairly similar ways in that they have autism spectrum disorder, and he has some mental health issues, OCD, anxiety, irritability, aggression, functional disease issues, constipation, fatigue, sleep problem, night twitches. In general, and then there's hypotonia. In general, these are extremely common to autism, and many people say, well, my child has some of that as well. So this is certainly not just autism, but it's relatively uncomplicated in that there are not any other severe disease manifestations, just the kind of neurological things that you can see in kids with autism. The sequencing that was done in PAM found a variant of uncertain significance in the SLC6A8 gene. This gene encodes for a creatine transporter. You may have heard of creatine, that bodybuilders use it because muscle requires creatine to store energy, but brain requires creatine as well. And if the brain doesn't have enough creatine, you can get intellectual disability, including autism, and sometimes seizures. And this really shows you as to how it's really a conversation between the genomicist, that would be me, the person who reads the genetic code, the physician that's caring for the patient, who's ultimately going to have to make the decisions and decide on the treatment, and then the family that knows what's going on with the patient more than anybody else and has to decide and has to implement that treatment. It's really the three of those people need to get together to really understand how the genetics affects that individual and family and what to do about it. So when I had, in this particular case, I remember there were 3 genes that I thought might be related to autism in PAM based upon the genetic findings. And when I discussed it with the father, who happened to be the ordering physician as well, on the first 2 that I thought they were the most likely ones, he's like, well, it really doesn't fit. But this third one, he felt it fit really well because you can see that, and this is the bottom box there is actually, I just cut and pasted from the report that was given. The stars I added in there. But the stars are areas that met. And particularly, he had these aggressive behavior episodes, and so he thought that this particular one was likely to be the cause. Creatine transporter, if you can't get creatine into the brain, you can give more creatine and hope that some of it gets across, but that doesn't always work. They tried creatine and it didn't work. So the father went ahead and got cyclocreatine, which is in some energy drinks. And cyclocreatine crosses the blood-brain barrier into the brain, and where it turns into creatine, but it doesn't use the creatine transporter, it just goes straight across. And you can see that some of the advantages here is that he had more language, less anxiety, fewer behavioral manifestations, less frustration. And so again, core areas of autism, language, behavior, were improved. Not because that there's some wonder drug out there, but because we found a particular cause of autism in that individual and addressed that directly. The next one's Kelly. Kelly is more of a complicated case. Carrie, she presented to my clinic as a teenager. She's now in her mid-30s. And she does have an autism spectrum disorder. This is her self-portrait. In fact, she has the artwork that I have in this entire presentation, including in the beginning slides, is all Kelly's. She's a very gifted artist, but she will not talk to anybody except her mother ever under any circumstances. When she wants to communicate, and she will communicate a little bit with me now that I've known her for literally 20 years. What she will do is that she has a computer and Siri's voice will come out, and she can type words on there in shorthand, and she comes up with entire paragraphs of speech. She's actually brilliant, but she will not talk, and she would not talk to anybody, or actually she won't leave the house in any way whatsoever unless it was a doctor's visit, and only then because she had to. But that wasn't her complaint. Her main complaint was continuous migraine, that it was 24/7, 365 with pain. She also had severe chronic fatigue syndrome, bowel dysmotility, dysautonomia, depression, and anxiety on medications. Biochemical testing, again, revealed signs of a mitochondrial disorder, which is, again, what I mentioned is common. And she did improve the physical manifestations, her fatigue, her pain, the depression and anxiety, they did improve on a mitochondrial cocktail. And these were 20 years ago were the cocktails that I was using. Now I just use my own blend, which has 33 different ingredients in it. Found 2 mutations in the glutamase 2 gene, which takes one amino acid, glutamine, to glutamate. And you can see the little cartoon in the upper right there is the biochemical pathways. And I know that's really complicated, but in the middle is the Krebs cycle that makes energy. And glutamate, which you see on towards the lower right, goes to alpha ketoglutarate, the little alpha signal and Kg in the Krebs cycle. So based upon that, we gave alpha ketoglutarate supplementation, which you can get over the counter in certain vitamin stores and on the internet. And then the email, the mother emailed the response below, which is exactly what she emailed me. And so Kelly improved quite a bit. She actually started showing her artwork online and started meeting with peers and everything, still talking in serious voice, but it made a big difference in her life. She's had some difficulty since then. She lives up in Northern California where there were fires, and that's had some setbacks, but in general, that her life is significantly improved on cofactors and also on the medication directed directly at the enzymatic abnormality or the DNA finding. So, and this just shows that, first of all, that I am a bit of a geek, that this is from science fiction, that any technology that if it's advanced enough, it seems like magic. And it does seem like magic that you can sequence 3 billion nucleotides. Even 20 years ago, this would have been total science fiction. 10 years ago, this is where I wanted to be, somewhere before I retired. And we got here very, very quickly, actually. And then my daughter loves to have her picture shown. So we're talking about sequencing the DNA. You really need to know about some of the difficulties in doing this to really understand why it's not done everywhere and why you just don't go to your pediatrician and says, oh, we'll just sequence the DNA and give you the result and we'll tell you what's wrong. First of all, is that there are 3 billion variants. There are 3 billion nucleotides, nucleotide pairs. So you get 3 billion from your mother, 3 billion from your father. And we sequence them all today. Variants can be small or large, as you can see on this here, and they can be rare or common. So if you look at this slide, most of the variants are a single base pair. They change one nucleotide to the other, and if it's in a gene, it changes one amino acid to the other. So that's on the far left. But you can have a variant that involves 2 nucleotides or 3 nucleotides or 100 or 1,000 or 10,000 or 100,000 or 1,000,000 or 10 million. When you get up to the millions, you're starting to talk about chromosomal abnormalities, something that you could see on a chromosomal test from years ago, if somebody remember what that was like. So on the very far left is where most of the variants are, just a single base pair. And on the very far right, you have chromosomal abnormalities, which are now found by microarray technology. So in microarray technology, you can find things from several hundreds to 1,000 to larger. And sequencing, you can find 1 base pair to around 50 or so. And you can see that there's a gap in there. And between the chromosomal abnormalities on the far right and the single base pairs on the far left, there's a lot of room. And we do find mutations that cause disease all the time there. In fact, I'm going to show you 2 cases in a few minutes. And until very recently, there's nothing we can do about that gap. But now whole genome sequencing, and that's one of the takeaways that I want to mention, is whole genome sequencing is really what we should be doing. We'll do everything. So beforehand, we used to do sequencing and microarray. And it's not like you can do one or the other, because they were completely, they did different things, and then they didn't find everything. But whole genome, we'll find 1 base pair, we'll find 10 million base pairs, we'll find everything in between. So it's the one test that can really do everything. And so the first recommendation is that, particularly in autism and in any of the neurodevelopmental disorders, whole genome is the way to go. Now, this is the machine that does it. For 10 years, it took to sequence the first person's genome. And it took billions of dollars in laboratories all over the world. This machine will now do it in an hour. It will sequence the DNA of a person, all 3 billion base pairs, and to sequence each and every base pair hundreds of times each so that we have very good indication that it's correct. The machine costs $1 million. And so that's one of the things about that is that even though the price of this technology has gone down dramatically, it's still not like in the order of hundreds of dollars or so. The price of this technology is still on the order of a few $1,000s, and most of it is involved in the machine cost and interpretation. So this talk is really about neurodevelopmental disorders. Yes, it's about autism, but it's also about any other sort of neurodevelopmental disorder, intellectual disabilities, epilepsy, loss of milestones. So what do I mean by that is they really have the same genetics. I know this is a very complicated slide, but if you look at that diagram, all of those genes are involved in autism, the whole diagram. But some of them also cause metabolic disorders, the blue. Some of them cause congenital heart disease, the green. Some of them intellectual disabilities, the red. Some of them schizophrenia, the yellow. Some of them epilepsy in the middle there. But there are many other things that can be added. Migraine genes overlap with autism genes. Enteric nervous system genes, like the GI, irritable bowel, cyclic vomiting syndrome, all sorts of GI dysmotilities. Dysautonomia overlaps. Depression and anxiety overlaps. Really, what these genes do, there are not autism genes that cause autism. There are genes that build a brain, maintain a brain, operate a brain. And if those genes have changes, if enough genes have enough changes, or if a change in one gene is severe enough, it interacts with the environment to predispose towards autism. And I mean predispose. Genetics is not preordained, even in identical twins, that you can have one with autism and one without. Genetics causes risk. Genetics does not cause disease. So there's two things I want to get to the slide. First of all is that, like I said, there's nothing that's preordaining. If you find the genetics, it tells you your risk. It doesn't tell you what you're going to get. The other thing is that these genes cause, or I should say predispose towards pretty much any neurological disorder and some non-neurological disorders like congenital heart disease. The importance of that is the presentation I'm talking about is really for all neurodevelopmental disorders because they're really the same genes in all of them. And so this is what I just said. Okay, even more complicated. This is a computer diagram of different people that with autism that have been put into different categories based upon sequencing and artificial intelligence looking to separate them out. And there are many different pathways involved. There is not one pathway that will lead to autism. I showed you before, I showed you a mitochondrial, I showed you a transport disorder, and there's many other pathways that will lead towards autism. And the importance of that is that many of these pathways are treatable. I took this out of the literature. It's actually a paper from 2016, which is actually at this point right now 4 years old, so it's really ancient stuff. But the blue arrows are mine. The blue arrows are pathways which are treatable. There are some treatable pathways. In fact, a lot of the pathways are treatable. So if you can identify the cause of autism, and actually today I can find it, I don't have an exact percentage, but I think it's around 75% or so I can find it, either the cause or a major contributing factors in patients, so it's not every time. There's a good chance that we'll be able to treat it because a lot of the pathways that lead to autism are treatable. Not all are, but a lot of them are. Okay, you saw this diagram before that, yeah, whole genome sequencing can do it all. But I just wanted to show you, I'm going to show you, at first, I'm going to show you some of the larger mutations. The 3 patients I've shown you so far, Kelly, Payam, and Carter. They had single base pair changes. So they were on the very far left of that. But I'm going to show you now a couple chromosomal abnormalities that are on the very far right of this. And then after that, I'm going to show you a few in the middle of some changes that are more towards the middle of this slide. So it's really all of these need to be looked at. So I put this slide together actually 4 years ago. So the CNV is copy number variance. It's too much or too little. And you can see the top one is 16p11.2. So that's really chromosome 16, the P arm. The P arm is the short arm of the chromosome. The Q arm is the long arm of the chromosome. And it's band 11.2. And at that point, there were 79 published studies that showed that abnormalities of that band are associated with autism. So that's how you read that. And there's actually thousands of copy number variants that are now associated with autism. So these are the more common ones on here. In the olive colored box on the bottom, those are the typical abnormalities of a large chromosomal, of a large copy number variant. They used to be called chromosomal disorders because you can see it on chromosome testing. So a large CNV will almost always have intellectual disabilities, other neurological defects, growth abnormality, usually small but sometimes big, birth defects, congenital anomalies, and dysmorphic facies, or they look kind of syndromic, which are really minor birth defects of the face. But that's what you can see with a large chromosomal, with a large CNV. Small CNVs, you don't see that. Small CNVs look just like a sequence. It can look like anything. So I want to show a picture of the third one there. This is what's called the DeGeorge region because it can cause DeGeorge syndrome. People know about DeGeorge syndrome generally because it causes cardiac or heart defects. It's the TBX1 gene that causes the birth defects. Not only the facial defects, you can see the nose and the ears in that child look different, but the TBX gene also causes the heart defects. If you have a deletion of TBX, then you're very likely to have a heart defect, about 50-50. It's like flipping a coin. So it's risk, again, doesn't cause it. But if you have a COMT gene abnormality, then you get the behavioral changes. Of course, if you have a larger deletion that covers both of them, then you have the birth defects and the behavioral changes. So it's based on the individual genes, even when you have an entire area knocked out where there's many different genes involved. So somebody may say, oh, I have a deletion on chromosome 2. Well, that's nice, but chromosome 2 has thousands of genes on it. Which ones are gone? Because each and every one potentially has clinical manifestations associated with it. If you have a defect on chromosome 22, if it's in the band 11.21, I'll say, okay, but does it involve COMT? If so, you might have behavioral changes. Does it involve TBX1? If so, you might have birth defects. So even in cases where you have a microarray abnormality, people say, oh, well, that's the cause of disease. There's nothing you can do about it. That's not necessarily true at all. Let me show you why. This is a case that I heard about a year ago. It was done in the peer-to-peer system, which is when the doctor and I were on the telephone, and the doctor explained the patient, and I explained the genetics, and we discussed how the two of them would meet. But he told me about a 19-year-old male that he saw because of severe selective mutism and social phobia. That this individual, he was known to have 22q11. And he had birth defects, a normal heart, but he had the other birth defects. And his gene defect covered both of those genes, the COMT and the TBX1. And he had some autism before, minor autism, but he was in school and he was, living a reasonably normal life. And a few years ago, he decided that he didn't really want to have any social life at all and to talk to anybody. And now he won't talk to anybody and he won't leave his room. And everybody's saying it's the autism, but this doctor knew. He says, I've seen autism all the time. This is not autism. There's something else going on. So we sequenced him and looked at all, and they didn't find anything. But they said, it's probably the COMT, because the COMT is risk for behavioral changes, but it's also a high risk for schizophrenia. About 1 out of 3 or so that have COMT deletions will get schizophrenia. And actually, schizophrenia really fits if you have a teenager who suddenly decides not to talk. That's actually more likely to be schizophrenia than autism. But because he had autism, everyone points to that. Some of you might have seen that before. It says, oh, that's just autism. Well, not really. You need to figure out why they do that. Next one I want to mention is the 15q one that can cause Angelman syndrome. With Angelman syndrome, you also get absent speech, but you get epilepsy, ataxia, and a bizarre laughing. What I mean by bizarre is that they'll just laugh at inappropriate times, and you can't really figure out why they're laughing sometimes, or they laugh instead of cry, or laugh instead of getting angry. This can be caused by a problem with epigenetics, so imprinting. So this is actually one of the situations in which you need to identify it and do a special test for Angelman, because even if you do whole genome sequencing, you may or may not find this because there are some imprinting errors. So this is pretty much the last holdout from an age in which we looked at a patient, we had to figure out what they have and test for it, where now we can do the whole genome and then go backwards to see what the patient looks like. So those were the larger copy number or the chromosomal abnormalities. Now I'm going to show you a couple of cases in the middle. So these are not large and they're not small, but they're sort of in the middle. First one is a 22-year-old, epilepsy, autism, intellectual disability, anxiety, and hypotonia. Well, that's a very common combination. In fact, 1 out of 3 people with autism have some degree of epilepsy. So that's not that uncommon at all. Singleton, that's just to do the patient, not to do the family when you do a singleton. Trio is when you do all of them. So we just sequenced the patient, and the official laboratory report found that, I'm sorry, it's this is not singleton. I need to change the slide. This is trio. We did all 3 of them and found there's a de novo variant. 64,000 base pairs, so it's not millions of base pairs on the X chromosome, and it included 2/3 of this gene here, ARHGEF9. This gene encodes for colibistin. Colibistin is a protein that's necessary to put glycine and GABA receptors into a cluster. Glycine and GABA are the inhibitory neurotransmitters. We commonly see defects in glycine and GABA in autism because they're inhibitory neurotransmitters. And if you have a problem with inhibitory neurotransmitters, then you're going to get overexcitation. Overexcitation can cause anxiety, autism, and epilepsy, migraine, all sorts of things that are causing problems because the nervous system is acting too rapidly or too overstimulated. So colibistin is needed to rope those glycine and GABA receptors into clusters. For them really to work, they have to operate as a cluster. So if you don't have colibistin, then you have impaired inhibition. And so you have an overexcited nervous system, which can cause epilepsy, autism, and anxiety. He also has hyperkyplexia, which is like you come up behind someone and say boo, and then they move around in an exaggerated manner. It's a very exaggerated startle reflex. It's hyperkyplexia. And I did not know that until I found the mutation. I said, does he, by the way, have an exaggerated startle reflex? And I'm like, oh yeah, you just come up to him and he moves around for like 3 seconds or so. He's very, very easily startled. And so that really kind of clenched it, that this really is what's going on with him. So he said, okay, well, what we should do is we should add glycine and we should add GABA antagonists. So drugs and in particular, dietary supplements, which improve GABA and include glycine. And unfortunately, that didn't help him. So sometimes you can find it and the treatment doesn't work. It doesn't always work, as the cases that I've shown you before, but at least we have an exact diagnosis. Now we know what the problem is, so we don't have to look any further. The next one is another medium-sized copy number. This is 84 kb or 84,000 base pairs. He has severe autism, intellectual disability, migraine, GI symptoms, and anxiety. Pretty much exactly the same as the one before, but with the addition of migraine. The brother is similarly affected, but actually he doesn't have the intellectual disability. He has sort of some autism, but he really is more normal intelligence. There's mitochondrial dysfunction by testing, which there often is. And but the laboratory said there was nothing wrong with the DNA. It was all completely normal. Now, when I took a look at it, I found that there was a de novo, meaning it's a deletion that's in the child, but is absent from the parents in two enzymes, USP17L2 and USP17L7. Now, the laboratory is not going to look at those genes because they've never been associated with disease before. But these are deubiquinizing enzymes. They're enzymes which tag proteins for destruction when they're old, and they untag the proteins if they say, okay, that protein's really not that bad anymore. It's like after a hurricane, someone goes through and tags all the houses. There may be a problem with these houses, don't go in it. And then they do that very quickly. And then later someone spends time and says, you know, this house is really okay. You can go in and get your stuff, or this house is not a problem at all. We'll take the tag off, or this house really, you can't go in. So this is a de-tagging enzyme. De-tagging enzymes are very important in neurodevelopmental disorders, particularly in autism as well. Angelman is a deubiquinizing enzyme. And because if you can't destroy old proteins, the proteins build up and they cause neurological disease. Because remember, your nerve cells last decades. And if you can't take out the trash, it builds up. For other cells that don't last that long, it doesn't really matter if you can take out the trash. There's going to be new cells to take its place. But in nerve cells, it really does make a difference. So with a deletion that was not in the parents, it's a new mutation in two deubiquinizing enzymes. This is a new disorder that has never been described before. Based upon this, it suggested to me that I should be using dietary supplements that will, as much as possible, remove things that the nerves need to remove. Organic acids, intermediates of fat metabolism, and I'm trying to think of in disorders of mitochondrial metabolism, you get free radicals. We need to remove those as well. So I put him on a cocktail to try to remove those as much as possible, and he did improve quite a bit. You can see on the bottom there, his transitions became better, sleeping better, using some new words, trying new foods and activities. She ran out and the behavior worsened and then improved again when she restarted the cocktail. So sometimes when we find the disorder, it works and the treatment makes a difference, and sometimes it doesn't. There's no guarantees with this that we'll find it, and there's no guarantees that the patient will get better. I will tell you that overall, about 50% of the patients improve when we do the DNA testing. Because some of the patients we can't find anything, some things we find things we can't treat, and sometimes we find things that we think we can treat, but it doesn't work, like the last patient. But about 50% of the time we find something is treatable and the treatment actually makes a difference. So that's pretty good. So no guarantees, but it can make a difference. The other thing is that we're finding new causes of disease all the time. In fact, I found one yesterday doing a peer-to-peer. At least once a month, we find a new disorder that's never been described before. It's because we have a DNA sequencer that looks at 3 billion nucleotides, and this is something that's really brand new. So it's really an uncharted area, and we are finding new causes of disease very frequently. One of the things I do want to keep in mind here is that autism is complex, and although I showed you a lot of cases in which there's a single gene which clearly caused the disease in some of these cases, it's really in a lot of cases of autism, perhaps in more than half, there are many different variants, some inherited from the mother, some inherited from the father, which produce the risk for autism. And so you can have variants, it may even be common variants in the population that are inherited from both parents, and the combination of those variants together is what leads to autism. That's really common. And a lot of times we don't find this is the cause, this is it, but I find candidate variants, or say, here's some variants that may or may not be involved. And here are some treatments that may or may not help those variants. So it's not always we find a single variant, but sometimes it's more than one variant. In all of the cases that we went so far, as I also look for other variants in those patients as well. So here's the same ones you saw before. In the past, we were looking at small to large, from left to right. Now I'm looking from top to bottom. On the top, at the very top is novel. That means it's never been seen before. It's not on any of the databases. And many of the mutations I've shown you are novel, that they've never been seen before. They may not even be in the parents. That's a de novo, which just means new in Latin. And if it's very rare, it might be causal. That means it's a very high risk. That means that variant, remember, genetics causes risk, but risk can be high or low. If it's very, very rare, it can be a very high risk, meaning that variant by itself can predispose towards disease. But the variants that are not very rare are not going to cause disease. In particular, at the bottom, you have common variants that are seen in a lot of people. That can't cause the disease, because why doesn't everyone else have autism? These variants are lesser risk factors. So really, we're talking about risk factors from the top to the bottom, but we're talking about the magnitude of the risk. If it's extremely rare, it can be a very high risk. It may be a very low risk as well. But as you start getting lower on there, as you start getting more and more common, then it's lesser degrees of risk. But that's still important. Factors that have low risks may still be treatable, and they still might be helpful in the clinical treatment. So here's another one here. It's de novo variants at the top. These are ones that are not present in either parent. You can see the underline. Inherited variants are inherited generally from one parent, but occasionally from both parents. The new mutations often are the main cause of disease, but the inherited variants, unless a parent has autism, they're not the cause, but they are simply a contributing factor, that contributing to risk. So what I wanted to show you towards the bottom of the slide, these inherited risk factors, you can be completely normal. The parent that it comes from is often completely normal. They can also have an autistic spectrum disorder, another neurodevelopmental disorder such as ADHD, but often they have what's called form-thrust phenotypes, and that's a very mild phenotype. Autism really is that the brain turns inward. I mean, at the neurological level, what's really going on is that the connections between different areas of the brain that are far away don't work as well as the connection between neurons that are very close to each other. And so we find a lot of things in building synapses and working on axons and the electrical grid of the brain. And it involves long distance communication. So really what you have in autism is you have the long distance communication is not working as well. And long distance communication between different parts of the brain is necessary for social response, and particularly language response. So that's what you see. But if you have a little bit of it, just a little bit of autistic tendency, you can turn in and not be really, the whole brain's not really working as one unit as well, but you can really focus on something. And a little bit of this really makes a big difference in people. In fact, it's very common in billionaires, and it's very common in people in the professions, not just in engineering, but in medicine and everything else. The ability to focus inward, to think, to be creative, to ignore what's going on in the background, to ignore your emotions and concentrate on it, is something that's very important to get through higher education, and also to be to be creative and to get ahead in your profession. So very commonly, a parent and other members of the family of an autistic child are simply not autistic, but you think of them as super people, and that they can do more than an average person can, because our technological society rewards that kind of inward thinking and ability to control oneself. So it's very often that parents will be highly unpaid professionals. And that really is what I call a form thrust. It's not that that gene is doing nothing in them. It's just causing a little bit of the effect, and that little bit of the effect can actually be quite positive. We're running out of time, so I'm going to go through a couple of cases here. I do want to talk very briefly on Dane here. He's had multiple episodes of regression. When episodes of regression often are mitochondrial or channelopathy, where the ion channels are not working as well. Remember, for the wires in the brain, they work by ion channels, opening for sodium, potassium, and calcium to go back and forth. So in Dane, we found that he has a de novo mutation in a GABA receptor. Remember I said that if you have a inhibitory receptor, that can cause overexcitation, and he has a de novo mutation that's not found in either parent. And so that's the main cause of his disease. But he also has a calcium channel variant that's inherited from the mother in the calcium channel A1S gene that causes periodic paralysis. And he has episodes of periodic paralysis as well, and cyclic vomiting like episodes. And every time he gets one of those, his autism gets worse and he loses milestones. He regresses. We gave him acetazolamide and potassium, which are treatment against the calcium A1S, and he's gotten much better. He hasn't had any episodes of regression since we've done that. And that's been maybe 6 months ago or so. And he's even gotten sick since then. So the GABA receptor defect, we've been treating that, and it hasn't been very successful as it has been in some other patients. But the calcium channel defect, which was not his major cause of autism and is inherited, was treatable, and it was impinging on his autism, and treating that made a big difference. So even if you find the underlying mutation, you can sometimes find more in the DNA that can make a big difference. Okay, sequencing. At the top, there is whole genome sequencing. You may have heard about whole exome sequencing, that's in blue. Whole exome sequencing, which is also called exome or WES for whole exome sequencing, is when you sequence all of the genes, all 23,000 genes. Whole genome is when you sequence the gene and everything between the genes. Now, only 2% of the DNA encodes for the genes. So when you're doing whole genome, you're really doing about 50 times the sequencing. So it does cost more. It doesn't cost 50 times more. It's generally about 3 times more or so. But there are a lot of mutations that are in the 98% of the DNA in between, and we're finding them every day. And when you do the sequencing of the entire genome, you get a lot of different things. At this point right now, I strongly disagree on anybody who does anything below exome. Exome is what most of the universities are still doing. Why they're not doing genome, I'm not sure. But panels in which you just do a few genes here, or maybe even 100 genes, a lot of the insurance companies want panels to be done. And some of them won't even do the larger ones until the panels are done. I very much disagree with that, because if you find something, it may not be the cause. You may have found something else. And even if you do find the cause, like I showed in Dane, you may not find what's really treatable. So I do recommend at least exome, and better if you can do genome. So next slide. This is an older slide. This can give you kind of some of the information. Every single line there, it's a different variant. The average person has about 1 million or so variants. There are maybe 10,000 variants that are in and around the genes that might be an issue, and the computer will look at all of that. The next slide here is, instead of the alphabetical order, these are the ones that I thought were important. By the way, this is Dr. Temple Grandin. This is her DNA. We've now sequenced her entire genome and found a whole bunch of stuff in her, including probably why she has autism that she has. There is a synaptic gene mutation that involves in building the synapse between neurons, but also found a lot of other things, like why she has missing teeth, why she gets hot flashes and pain in her extremities, why she has rashes, and several other things that we've found in her by doing DNA testing. Genetic counseling. Before you do testing, obviously that you should talk to the doctor about what it can find, what it can't find. What does a positive result mean? Well, most of this talk has been about a positive result. What does a negative result mean? Negative result doesn't mean you don't have autism. That's ridiculous. A negative result doesn't mean it's not genetic. It's just today we can't find it. Many patients have been sequenced. I didn't find anything. And then later, with more knowledge, I went back and said, well, this is what's going on. So once you have the entire sequence on a computer, you can always go back and look at it. And I do that with my patients, particularly the ones that are not doing so well, or if there's something new that come up, it says, well, what's going on here? Let's look back at the sequence. There's on-target, off-target findings. There's all sorts of things that we can find when you do DNA. In particular, you can find increased risk for developing disease, carrier status, and that might have reproductive interest, consanguinity. Are the parents related? A lot of times the parents say no. I find otherwise. In fact, about 1 out of 3 of the people that I look at, the parents are related. Yesterday I identified 4th cousins. It's really common. Most people in this world are related to their spouse because DNA can find it. Think about all of the, go back 10 generations or so, about every single, and your spouse 10 generations. Yeah, it happens. But when you get beyond 2nd cousins or so, really the risks of causing trouble from that are really, really small. Misattributed paternity. When we first started doing DNA testing, once in a while we would find that daddy is not really daddy. I haven't seen that in 5 years or so, mostly because women in which that might be an issue probably are not getting genetic testing. But anyway, that's a potentiality. Especially when we do triomes and we're doing both parents, obviously I can tell. Disclosure options for families. There is the option to only know what's causing the child's disease and not know additional problems. I usually don't agree with that. The last 100 families that I've done, 99 wanted to know everything. 1 didn't want to know, but when that one family, when I actually had the results, the mother asked for them. So the vast majority of my families want to know everything, and I almost never find things that are really like nasty or something like that. It's really unlikely, and I'm not looking for things that are causing trouble. I've been doing genetics now for 30 years, and I haven't had a single family that said, I wish I didn't know something. That day may happen someday, but it hasn't happened yet. Okay, just a briefer for a second. Not all mutants are bad. These are my little mutant chinchilla babies, taking different strains of chinchillas that have genetic mutations and crossing them. I'm trying to make a more domesticated chinchilla that's very friendly. I have very friendly chinchillas, by the way, and I'm trying to make them even more friendly. Baby Waffles Jr., the little one there, is only 4 hours old. So not all mutants are bad. Oh, Baby Waffles Jr. is wild type. That's a normal chinchilla. The others have color mutations. And so anyway, is that in autism, there is strength. Many of these kids have quite a lot of strength. Others are very severely affected, and it's hard to find the strengths, but often they're there. Okay, pricing. To do whole genome can be expensive. Exome, where we do all of the genes, I can get it done well for as low as $1,000. And then mitochondrial DNA for $500, and then if you want to do a microarray for another $500. So you can do them a la carte for around $2,000 or so. It doesn't give you everything. Self-paid genome prices are as low as $2,800. The best lab, there are a lot cheaper labs, but you get what you pay for. The best labs have much better sequencing. It's much easier to interpret, much less likely to make an error, and I can find mutations much easier because of their computer systems. Most laboratories will take insurance. I look for labs that run insurance, but also I look for labs that will do cash prices. The labs that I use, they'll take insurance, and if the insurance doesn't go through, they'll run the cash price. These prices are not insurance prices. Insurance company is charged a lot more than this. These are the prices that you will pay if you need to do your credit card. There are some things that will help, like a good letter of medical necessity, appropriate codes. This goes over some of the things of the letter of medical necessity. These slides are available. I'm not going to go through this now, but a good letter of medical necessity as to why your child needs DNA testing and that it's going to be used to determine treatment. Often we'll get situations covered. Life is not fair. PPOs usually will cover autism if with a good letter and with good codes, maybe 3/4 of the time, maybe a little bit more if a geneticist orders it. HMOs is and government insurance, it's rare that they will cover, but it happens. Testing recommendations. I do recommend whole genome sequencing. Whole genome sequencing will give you microarray. It will give you sequencing, not just of the exome, but sequence of the entire genome. It will give you mitochondrial DNA. If you do whole genome at the right lab, it will give you mitochondrial DNA. It gives you microarray, better than microarray. It also gives you Fragile X and pharmacogenomic testing. I hate to give psychotropics without knowing if that will work and what dose to give, because you can look at genes to say, okay, something like Zoloft I use in autism very frequently. It helps with the kids' anxiety. It helps with their social ability. But not everyone will get better with Zoloft. I can look at the genes and tell you which ones will likely get better and which ones will not. And then I could look at the genes and say, okay, you need a higher dose. We'll start at the regular dose, but then we'll double it very quickly because your liver will eat it up really quickly. And if you don't give a high dose, you haven't really tried the gene. You haven't really tried the drug. On the other hand, somebody may have the STAR4 mutations, and he said, okay, just a regular dose of Zoloft is likely to cause a lot of side effects. Let's start at 1/4 of the dose. That might be enough. Then we'll go up to half the dose. And then you can get good effects on lower amounts. It doesn't mean you can't use it. It means that it's cheaper because you use less of it, because that liver will break it down really slowly. So you can get that information off a genome that you really can't get off of exome. The other thing is trio versus singleton. It really does matter to sequence the parents. There are many different things that you can tell from that, but mostly it's de novos. De novos are common in autism. I'm finding about 40% of the time. That means with somebody with significant autism, epilepsy, birth defects, or significant intellectual disability, it's very likely that there's a de novo mutation that's causing it. You will not find it unless you sequence the parents. It's unlikely I'll find it otherwise. And you really do need to sequence the parents. Trio costs are generally 1.5 to double the cost of singleton. It's not triple the cost. And we're not sequencing the parents' DNA to find out all of their problems. In fact, you won't find the problems in the parents. The only thing that we'll ever find in the parents, if it's in the child, and then we look at the parent to see if the parent has it as well then you're going to find it. But the parent is not being looked at to see what they have. There's a lot of advantages of trios. That's where you do the parents, de novo. We can also look at recessive variants following the condition for the family. If there's families that have like Ehlers-Danlos type thing, or have a lot of ADHD, or maybe there's somebody else in the family with autism, we can find the gene and see if it's going down the right part of the family and what we expect it to see. And again, it said double the price, but some of the labs are coming down to 1.5 times. So here are my recommendations for trio: moderate to severe cases of neurodevelopmental disorders and or birth defects. I like to do trio in everybody, but it's really pretty much mandated in the situations of moderate to severe cases. In like an Asperger case, we don't call it Asperger anymore because Dr. Asperger was a Nazi, but in mild cases of autism, sometimes we can get away with just doing a singleton. Only your physician can determine what test is right for you. Unless I have evaluated the chart or something, I cannot recommend the testing, but I do recommend, as a general rule, trio whole genome sequencing. The key is who will interpret that. So the peer-to-peer counseling system is really where we connect the doctor, hopefully yourself and myself together, where I do the genomics, your doctor knows the situation, and if you're in a state where I'm licensed, and those you can see the list of them there, and like in the first paragraph in the middle of it, then I am licensed in New Jersey and Pennsylvania. then you can be on the phone as well. I can look at the child's face. I can ask questions about the family history. You can ask questions about what I just said. It really makes a big difference. You connect the family, the child, the treating physician, and myself, the genomicist, to go through and understand the genetics and to understand what the options are to do about it. And that's what really the peer-to-peer system is. So we can help your doctor to order the right test, to get negotiated prices, so it'll be a little bit cheaper for sequencing, and that we can interpret it and then discuss with your physician how that could be treated. And then this is the telephone numbers and emails and all of that down below. And Lori is the contact. I know that I'm running out of time, but real briefly, we are looking at the lab. We did 100 peer-to-peer so far. We did present the first 17 at the American Academy of Neurology, that they were 8 different physicians in 7 states and 3 nations. You can see that, I mean, there were only 17, but 16 of the 17 altered the physician's management, that based upon what was said, that different things were done. 9 of the 17, half of them received a diagnosis. This is what your child has. But the one in purple is the important one. Changes in therapy were 15 out of 17. We're trying to put this together now for the last 100 patients. Our understanding of the data is that it's probably 50% get an exact diagnosis, and 50% have significant improvement. Those are not the same 50%. Sometimes we have a diagnosis and nothing will work on it. And sometimes there isn't a diagnosis, but there are risk factor genes, and the risk factor genes are treatable. So it's about, it's about 50% get a diagnosis, 50% improve significantly, but they are not, they're not the same 50%. You can get a diagnosis and not be treatable, or you can not have a diagnosis and just have risk factor genes and be treatable. All 4 possibilities. You can have a diagnosis and be treated, or you can not get anything and not get any improvement. All those are possibilities. And you can see outside consultations, 4 out of 17. The insurance companies are afraid that when we do this, we'll find a million different problems and you'll end up getting a bunch of tests and everything. The additional testing, 41%, that was almost always to look at the parents because we did not do a trio. The next most common test was an EKG. Nobody got invasive testing, no surgeries were done because of the results, and the indications for outside consultation, that's what's usually, sometimes it was a cardiologist, sometimes it was developmental pediatrician to better treat the autism or something. The fates of ancient Egypt, that it doesn't matter what the hero does, that their fate is written and they're going to die when they're going to die, no matter how good they are. That's not the way genetics works. I think about genetics as that we're all dealt our hand, like in a poker game. Some hands are better than others, but it really depends on how you play your hand. A good poker player can win with a bad hand. A bad poker player cannot get very far with an excellent hand. Genetic testing is looking to see what your hand is, to play your hand the best that you can. So know your potential, get tested. It's not going to tell you what's going to happen when you're going to die or anything like that. In fact, a lot of times people wanted more information. They say, okay, now that you have the mutation, how is my child going to do? And I say, no, you're going to tell me how the child's going to do. Genetics can't tell you prognosis. I might be able to tell you what treatment to do and you may do better, but it's really not going to give me that information. How do you improve? Right therapy. The right therapy may be alpha-ketoglutarate. It may be cyclocreatine. It may be a drug. If it is a drug, which drug? Instead of just using all the different drugs, which drug should you use and what dose? Should you start at a low dose or a high dose? All of this information can be determined many times from the genetics. And then here's the last slide. I will leave it on here. And that's, in particular, all of this has been recently published. If you look at the middle there, I know that is the hyperlink. It doesn't work from your screen probably, but to go to the article in which it describes in incredible detail genetic testing in autism. This is meant for if your physician says, well, I want to know more, I don't want to just test, you say, well, here, read this. And then I'll let it go to questions now.
Thank you so much, Dr. Boles. Yes, we do have a bunch of questions. Some of the questions are a little specific. So those of you with specific questions, if you can message me or contact information, I'll look it up and I will send those questions over to Dr. Boles. But we did get one repetitive question from a couple of individuals, and it was about how often to get the genetic testing. So one individual got it done 10 years ago, another 20 years ago. Do you recommend that they get the genetic testing again?
10 years ago was in the dark ages. 20 years ago, I can't imagine what they would have done, just Fragile X. Yeah, even in patients that had whole exome sequencing 2 years ago, I recommend to go to whole genome because you can get so much more information. In the past, not that long ago, a few years ago, every year the testing would get so much better. Every year your testing was inadequate. And after 2 years, we would repeat the testing. And I've had a lot of patients that have been tested 4 or 5 times. And I know that's really hard to do for many reasons. The insurance companies don't like to do it. But now we're sequencing whole genome. There's not anything more that we're going to sequence. We sequence all of your DNA. So it's not like tomorrow we're going to have a better test. Tomorrow we will have a better interpretation. But the test will be, all of their sequence will be on the computer, and the interpretation can be done tomorrow, next year, 2 years from now, but you won't have to retest. So yes, if you haven't had whole genome, I recommend it. If you haven't had trio, all 3, I do recommend it. Even if you've had whole genome for the child, but not the 3 of you, there may be things to find.
Okay, great. Okay, so I will also send out an email to all of you that attended the webinar today, asking if you would like for me to share your information with NeurAbilities and Dr. Boles, so you can be put on their mailing list or email list, so you can receive up-to-date information about what they're doing at NeurAbilities. Thank you so much, Dr. Boles, for your time today. I know it's a bit crazy in the world, so it was nice seeing pictures of your chinchillas and your daughter.
Oh, you're welcome. Thank you
Okay.
for this opportunity.
Thank you so much. Stay safe, everybody.